How are the waypoints chosen for this dive?
Yeah. Can we zoom in on that crinoid?
Sure thing.
Yeah. Thanks.
All right, Dave, you can push it there.
Oh,
It's
it
a
looks
sea
like a
star. Oh, that looks
brisingid
like,
or something.
is that is it crinoid? I guess. You sure?
Can you zoom in on the center? It looks like a brisingid sea star that has
like a
lost
sea
some
star
arms.
to me, yeah. Maybe we should go for that.
Or has weird, I mean, yeah.
Yeah,
It's not a
new one.
crinoid. I don't know why it has the weird arms
It was
sticking out.
little. Yeah, there's a crinoid over there. That thing under the ledge. It's yellow. Yeah, this would be a good one to collect. We only have one echinoderm so far.
The ship will stop in 10 meters, so.
Okay, perfect. 10 meters ahead. All right, let's set up for this. That's a good question. It's probably a bit too big for the slurp.
Yeah, it's actually pretty big. It's like 40 centimeters across,
yeah.
huh? Is it? It's like pretty massive.
It is big. Yeah, that'd be great. I don't know, grab it and drop it in the box.
You could suction and drop if you're worried about grabbing it.
Drop, yeah.
Yeah.
What do we have in the front box?
We've got a decent bit in the front box, so that's a good thing to consider. Do you feel okay putting this in with things that are in either of the two front boxes? Yeah?
Yeah.
Okay. We've got 2 sponges in the left, and we have 2 corals on the right, so up to you which you think is better.
I mean, we could try suction into the front box.
You could suction lightly and see what happens. Yeah.
So that's at 440 meters.
Let's try. Okay.
May you tilt the camera down?
Yeah. Just give me a... sorry, can we zoom out of Atalanta cam? Sorry.
That is big.
Thanks. Yeah, no, it's okay.
About that big.
Terrifying you?
Yeah. Yeah, let's just do like a light slurp.
Okay.
See what... It's gonna be really big for the
Suction's
front
on,
box, but...
and it's only at 10%.
Okay, you wanna tilt up a bit?
Yep. Here, I'll give you another view.
Oh, perfect. I'm gonna go for the center.
Okay. More suction.
Yeah. It's gonna curl in.
It's really hanging on.
Is that 100%?
That's now 100%.
Is that 100% on the...
Yeah, that's 100% on the suction.
Okay.
Nothing?
Yeah. So...
You kind of knock it sideways, maybe. Get it loose, I don't know.
Yeah.
Yeah.
Hey, buddy.
I gotta bring up the jar too. Oh,
Maybe.
Huh. I wonder if we clogged
like-
it at all. It seems not to be sucking as hard as usual.
maybe if you
I'm trying to get on this.
got like a leg or something
Yeah.
down the slurp and maybe
Yeah,
it would
maybe
stay
that.
there.
Here, do you want to turn
Yeah.
the slurp off and I'll try to get it lined up.
Oop.
There. Go down in the...
Let me know when.
Try that. I think I've got a leg in there. Yeah, thanks, Dave. I'm gonna try the center again.
Oh, almost.
Well, I've got a whole leg in there. He's not interested in... Oh,
Oh. Oh, it might happen.
oh.
All right.
Alright.
All right.
Well, let's try to get it in box.
Yeah, let's come wide. I'm gonna rack back the camera. Open the
Yeah.
box.
It's very sticky.
Oh, it's huge.
It's massive.
Nice.
I think
me know when
this
you
will
want me to-
be...
I'm gonna kill suction.
Yeah, kill section now. See if I can
Wiggle
get
it
its
free.
leg out.
Yeah, great
There
job.
we go.
Nice.
All right.
Nice.
Beautiful.
Cool.
The one leg sea star grab.
That was fantastic.
I
Yeah.
can't believe it fit in the...
Forward box closed.
That is sample number 7.
Stow that.
Thank you, Data.
Thanks for your patience, everybody. That
Nice job.
was a very Velcro-y little...
Yeah,
That was a tough one.
we got all the legs. Let's
All right, racking on the camera.
Wait, did you say 7?
Yes.
Thank you. Can you see HiPAC and tell me if that's the correct way of doing sample numbers? Oh, never mind.
Yeah, I don't know if
I'll go
we
back.
usually do with the dash.
Roger. Thanks. It's been a while.
You're all good.
Actually, HiPAC looks like an underscore.
What do we have in sample jar
No,
1?
you did it right. I think that was right.
I'm not sure.
Data? What do we have in sample jar 1? Do we have anything?
Sorry, what do we have in sample jar 1, you said?
Yeah.
It was coarse grained sediment.
Okay. Cool.
Oh, you were saying we were doing underscore before, Samantha?
I'm going to rotate us back to the flush jar. So, you're gonna...
Watch out, the sediment might swim out.
Yeah.
Okay, put in a move if you're good. Jake, good for move? Great.
Yeah.
Bri, good for move?
Yeah.
Perfect. Bridge nav. Three 0 meters, 085.
We have a question from the chat asking about why this area might be more sparse in terms of organism abundance.
Nothing like a simple question. You know, just gut feeling looking at it, you know, there's really not much accumulation of sediment, and that would imply perhaps that the slope is a little too steep for like organic material coming down from above to sit on the bottom and then be accessible to organisms that are living here. You know, we saw a lot of things that live on and within the sediment down deeper, and you know, they just don't really have a habitat here maybe for most of them. So I would suspect it's a combination of those two things. Oh, look at that, the crinoid there. Can we stop and have a look at that?
Yep.
Yeah, we've been
Yes.
seeing a lot of these
Yep.
isocrinids.
Push in
Okay.
there.
That would be my gut feeling, you know. Beyond that, I really can't say much. Just guessing at this point.
All right, you happy there?
Thank you. Yep.
Come on.
We got a cucumber like
I know.
bananaing on the top.
I might point out that looks like from the bathymetrics we're coming into an area pretty quick here, which will have a steeper slope too, so that may affect what's coming down the slope from above. It's interesting, you know, we really haven't seen any fish in here, have we?
Not
in
a ton.
This zone? Yeah,
No.
look, I mean, it's great fish habitat. It's got a lot of relief and cover and all that. Maybe there's nothing to eat.
Oh, basket star. We haven't seen one of those yet.
Yeah.
Would you like a zoom?
Let's have a zoom on it,
Okay.
yeah.
Yep, push in there.
Certainly don't want to collect it. So we're 420 meters. Yeah,
Thank you.
All right, come on.
okay.
Oh, you were so quiet.
What about that?
What do we have there on the right?
Sideroid urchin, Laurie? That the one you wanted? No. Okay. Let's zoom in out real quick,
Okay,
Deb, and
push
then
in.
move
Cool,
on,
snap
I
zoom.
guess. Yeah. Okay. That's good, thanks.
Back on wide.
Sometimes it kind of feels like we're playing Pokemon.
Gotta catch them all.
Yeah, this is a nice little C-star. Can you zoom in on that?
Sure thing. Push in there, Dave.
Yeah, we're checking the book, see if we've seen that one before. It looks promising. We'll just maybe hold, and we'll decide whether or not
Sure.
to collect.
Help.
Yeah, we'd like to collect that one, please.
Roger that.
Hold position, please.
Don't think that one will fit in the slurp either.
Yeah, I think we're looking at putting it in the box,
Yeah. Do we want to
slurp
grab
and
it,
drop.
maybe?
Another forward box
Yeah.
sample. If
Yeah.
it's okay, that put it in there with the other
Yeah.
samples. Yep. Okay.
Okay. Which side is better? Should have checked last time. Do you have a preference for which side of the forward box?
Not really.
Okay. Cool. I just realized I didn't check last time. I should have. But
It was in the port side last time.
okay, let's see where it'll fit. I feel like start with slow suction because
Yep.
don't want it to get sucked in there and stuck. Okay.
Yeah. Been saying we need lights that go like look up like that. Yeah. Pick it up. Would you like me to tilt up?
Yeah. Let's tilt up a little bit. Thank you. And yeah, I can probably do it. Yeah. Okay. Let's try suction on low.
Okay, it's at 10% now.
Okay. All right.
I'm gonna turn it on.
Yeah. Maybe 20.
Okay, 20. It's moving. Oh,
Eh,
30,
Yeah, 30 or 40.
40.
Let's try that.
More beans. Oh,
There we go.
perfect.
Oh,
Ah.
A little more beans.
more beans.
At least it can't hang on, with that.
Yeah.
Okay, 50,
Let's just, yeah.
60, 70.
I don't think it'll get sucked in. Yeah, okay. Nice.
Okay,
And
rack.
we retracted.
Racking back the camera.
Nice. Okay, good for front box to come
Okay,
out.
front box open. Oh. Wow.
And you don't mind which one?
Nope.
Okay. I'm gonna let's turn suction off. I think it'll just
Suction's off.
fall in there.
There's a fish.
Suction is off?
Yep.
Okay.
Oh.
There we go.
Well done. Well
Another
done.
one. Forward box closed.
Things that sink are fun.
That was at 410 meters.
Alright. The arm away.
Nice. Alright. Onwards and upwards.
Onwards and upwards.
Bridge nav. We can do another 30 meter 085.
I'm going to zoom in on that
Right, white
coral here.
Yeah.
This, yeah. Just quick zoom. Another one, that same sea star we just got too above it.
You can push in there.
Okay, that's that coral we got deeper. Yeah, or tried to get. Yeah. Okay, that's great.
Okay.
Thanks.
Come on. That's the one that fell apart when it was touched. Very brittle, right?
Yep.
Yeah.
That's that same sea star we got just a few minutes ago. There's a couple of little assisted sponges, it looks like. They just keep on going though.
Yep.
And we just crossed the 400-meter barrier. Above 400 meters now. Why are there no fish? Very interesting.
Are we going to be taking any Niskins on this dive?
No Niskins.
Bridge nav. We can add another 30 meters to this heading.
Those fish.
Another one
Yep,
of those
there's a fish.
What
Got
were
a
these called?
fish.
Boarfish?
Yeah, it looks like the one we saw earlier.
Yeah, let's we zoom in on him.
You can push in there, dude.
Ooh, lovely.
What kind of fish is it?
Boarfish. seen that before, but yeah. Interesting. Great. Okay, we can move on, I guess. He was at 390 meters.
Are those shells or?
Looked like shells, yeah, probably,
Yeah.
you know,
Huh.
well down from above. Can you zoom in on just for
Yeah.
for a
there.
quick look? Yeah, could be an octopus, you know, nest. There's a crab carapace to the right. But yeah, it kind of looks like maybe an octopus has put those there or something.
Squat
Hard
lobsters
to-
in the crevices? Look like
Are
too.
there?
Oh, yeah.
Yeah. Okay. Thank you.
What?
Interesting. Oh, and maybe some glass sponge in the middle there too.
Yeah. A whole bunch of stuff.
Yeah, interesting.
We have an ID of that fish from Megan Putz. It's possibly Hilardia ghostline, which is a Hawaiian spikefish.
Hi, Megan.
I thought Megan was on vacation.
Yeah. What are you doing, Megan?
Megan's always watching.
There's that same sea star again, I think. Yeah. Can we zoom in on these corals real quick?
The yellow?
Yeah.
Yeah. All right.
Yeah,
You push in there.
Just get a look at them.
What is that?
The,
Yeah. Yeah. Lovely.
I love the color.
okay.
Okay, great. Thanks.
What?
Onward and upward. That was at 380 meters. So we've sort of moved into that area with a bit steeper slope. Sure not a lot of organisms showing up. 370 meters.
Bridge, no? 30 meters, 085.
For folks who just tuned in, right now we are doing our first ROV dive of this expedition at Shark City in order to survey deep water coral communities. I feel like community might be a strong word here, though.
And have a look at this urchin real quick.
All right, Dave, put her there.
That looks like a different one. Does that look familiar, Lori? How many kinds of... I think we should go for it. Let's collect that guy if we can.
Okay, yeah, come wide, Dave. I'll set up.
It's got those, orange ambulacral grooves. Different.
Are we gonna do
Not
another
sure.
section then?
Yeah. What are we gonna do with this one?
Bridge, nav. Hold position.
So, I don't think we can grab it. It'll be too fragile.
Yeah, try this sucker.
Yeah, which means we gotta do what we did the last couple samples. I'm just bringing Outline up because we're kind of drift over you, so. So, yeah, are you guys okay if it goes in the front box again?
Yeah, I think that's okay.
Okay.
Yeah.
None of these organisms are going to eat each other or anything?
I don't think so. You never know.
Sure. You can do it now, actually. Yeah.
Any possible ID on this sea urchin?
Alright, yeah, let's, I mean, full suction, I feel like.
Full suction?
Or you can do like 75 maybe.
Okay.
I just think it'll be hard to suck with the lines. We got a small little.
Okay, that's 100.
Yeah, that's 100. Okay, I'm gonna try knocking it off a little bit first.
Nice.
Just gently.
The knock and slurp.
The knock and slurp. That's full suction.
Yep.
Almost like its spines
Wonder if
are
you have
meant
to,
to.
like, maybe flip it over or something?
Yeah.
I don't know if you can do that, but...
Let's see what's on the underside.
Did it tilt up?
Yeah, great.
Totally don't.
So try to get it against the rock, maybe. Can we go a bit wider? I just want to see more of my arm. Bring it down. Oh.
Yeah.
A question for the scientists. How do you distinguish deep sea corals? This is a relatively shallow dive compared to what our ROVs are capable of. So where do you draw the line between, I guess, shallow corals and deep sea corals?
Okay, let's tilt camera down.
Yep.
And then Dave, you can push in again.
Well, but shallow water corals have symbiotic algae, and they need sunlight to for those algae to photosynthesize. So that's a pretty good way to tell from a quote unquote deep water versus a shallow water coral, whether they're azooxanthellate as we call it, or if they're zooxanthellate. So if they have symbiotic algae or not. So, with light penetration, clear tropical waters, you won't find zooxanthellate corals deeper than about 90 to 100 meters. I guess the deepest sort of shallow water reef corals have ever been collected are about 120 meters, 140 meters off Hawaii. That's very, very clear water.
Yeah.
And then below that,
Flip it up.
they lack zooxanthellae. There are some species of coral, interestingly enough, who can have their zooxanthellae in shallow water, but then if they go deeper, they can still survive, but they lack zooxanthellae and probably grow much slower.
Oh. Here, I gotta repick the thing.
So,
I might
anyway.
actually try flipping it with the jaws.
Without the zooxanthellae,
Come on, Dave.
how do the deep sea corals feed?
I think, you want to just grab it.
Could do. We
Yeah,
do
give it a
risk
try,
crushing
but
it.
Try
I
to
think,
grab some of
um,
its spines, maybe. It might just come up. If
Yeah,
you grip
I
for
might-
zero and grab
yeah.
some of the spines, maybe?
Just the spines, no, that wouldn't help. But if you could kind of, even if the test gets crushed, if we can get pieces of it, they can use that to identify these
Okay.
surgeons.
I'll try to grab it. Kind
But
of.
if we don't get it with the
like
the
this.
grab here, we should just give up, I
The
think.
rock is really crumbly.
Yeah.
I'm gonna kind of bop it out a bit though. There we go. Okay, tilt down.
Tilting down.
And then let's try to. This jaw setting's funny. It's like full beans.
Full beans, but no beans at the same time.
Yeah, he's taken off.
Yeah, it's going faster than my jaws are closing. Alright, let's see if we can get him there.
Slipped out.
Oh.
Oh yeah, he's under the...
All there?
I'm all there.
See, it's right there. How far did it go?
How far could it go?
Oh no, did it roll all the way down?
May have.
Well, I think
There
we
you
should,
go.
okay, give it another shot.
Yeah, it's in a better spot. It's not up
Yeah.
against a rock.
But look at
here.
how fast he's going.
Yeah, he's really cooking.
he's really
These urchins cook. They might
Yeah.
move.
running for his life, so to speak.
Alright.
Poach out here.
Oh, sorry.
That's right.
My jaws don't,
Oh,
wow,
this
oh my
fast.
gosh.
He's mostly falling down the slope now.
Okay.
Oh, all right.
I think we should give him a win.
Okay.
Okay.
Take the L.
Win some, lose some.
Take the L.
Yeah.
Sea urchin, 1.
Yep.
ROV
Us,
0.
0.
Got good images of it. Yeah, if it's a known species, we'll
Urchins are tough.
be able to figure out what.
Evolution. Those spines.
Alright. There.
That was at 360 meters.
I'm gonna turn suction off.
Oh, yeah.
Alright, coming down. So you can have more leash.
There's a little fish. Eel. It's fast. Pushing maybe.
Sorry, there was a
Oh,
looked
you
like
wanna-
a sea star up there on
There's a couple of them.
upper
Sorry, Dave, you can hold there.
upper right
Okay.
corner. Yeah, that one. That's a good one. That's the one we want on the bottom. There's one up at top, one at the bottom. There's another one.
A whole bunch of them.
Yeah,
Oh,
that's a
there's sea
really
stars?
cool one. It's got like, this always reminds me of a pie.
Yeah.
You know how you make a pie and you make slits in the crust to keep it from blowing up?
Alright, you can push
Look
it
at the
there.
5 little slits on the aboral surface there. Huh? Looks good enough to eat, doesn't it?
Wow.
Yeah,
That's a
cool-
great one. Yeah, we want to get one of those. Yeah.
Okay.
Do that.
Okay.
Yeah.
Come on, Dave. we'll
You don't want
set
it?
up.
Wow.
I don't know. They're nice specimens. How many echinoderms do we have? We have 3. Well, let's get some good imagery, if not,
Okay.
nothing else.
One more push in, maybe? One more zoom? Thanks, Dave.
What's the ID
Yeah.
for this guy?
That's as far as you can zoom. Oh, that's nice. Okay. Yeah, so there's another one out to the right. If we just kind of have a look at each one real quick.
Sure, up and to the left.
It's like almost like a suction cup onto the rock. You see, like this one here, it doesn't have the edge of the animal flared out. Okay.
Okay. Little squat lobster.
Getting dragged around.
Yep.
Yeah, those are at 360 meters. Yes, another one up there. If we zoom in on that one on the right
Yep.
side of the frame, yeah, just a quick zoom in, just
Get all 3.
yeah, it looks a little, they look each one looks a little bit different, sort of
Okay.
depends on how that edge is flared out, I think.
You want to push in there?
Yeah, just Okay. That's a really lovely creature. Great, beautiful.
What were these ones called again?
Sorry?
What were these ones called again?
What's left? Got it in the book.
Come on.
What is it? Okay. It is a miracle. All right. We're onward and upward.
Yeah.
Those 360 meters. The first time we ever saw that, we couldn't figure out what it was, and finally, this guy at the Smithsonian, Chris Ma, finally was able to identify it. It's pretty common around the 300 meter level. Probably see a bunch more here, but we'll soon find out. Okay.
This is neat. This is like, a lot of this is very different from what we've seen at other sites.
Yeah. Got a squat lobster in the upper left. I'll zoom in on him. Oops.
Push in there, Dave.
Yeah, nice.
Oh.
Oh.
Yep. A lovely image. Great, I think that's good. We
Good.
can, yeah, onward and upward.
Do we have a ship moving, or?
I was just waiting for the ship to find a good heading again.
Okay.
Yeah.
Sounds good.
I'm gonna put one in right
Moving
now.
around.
Yeah. I think they're good. Bridge nav.
Cool.
When you're ready, another 30 meters, 085, please.
Okay, let's have a look at that real quick.
There's also a
Where's
fish
that
right in the
fish?
middle of the, yeah.
Push in
Oh,
there,
fish, there we go.
Dave. Yeah. Look at that.
Oh yeah, that's a paraperson. Cool.
It's really
I'm
pretty.
just gonna look
Yeah.
down at the tether.
That one, I don't know the name of it. It's been described, but it's a great image. Oh, wonderful. Yeah, can you just zoom back just a tiny, tiny bit to get the whole fish in the image? Yeah. Oh, okay. He's
Take off.
being a little bit cooperative. Nice. About 20 centimeters long. Okay, great.
Okay.
Yeah,
Onward.
onward. I noticed just where we saw him, there was a dead barnacle shell on the rock there. It's interesting.
Okay.
It's not a big deal,
We
but
might just have to wait to catch
Yeah.
up a bit.
we haven't seen any live barnacles. But
I feel like that fish was a really good example of having eyes on top of its head as a creature that seems to follow the bottom of the ocean.
yeah, so here's the guy we wanted to look at
Yeah.
before. Yeah, oops.
Just waiting on the
Yeah.
ship to get moving.
Okay.
Falling off. Fully stretched out with the tether.
While the ship is starting to move, might take a minute just to kind of go over future planning. I was looking back at how far we've come since the dive started. We've been averaging about 220 meters an hour, and at that rate, we've got about 1,600 meters left, and so that puts us at like 7 hours to get to the end of our waypoints. So kind of curious if we are interested in kind of speeding up this ridge to get up to the top, to the summit,
think
given that
that's-
we are
Yeah, I think that's
3
the
hours,
preferred
3.5.
option, to just move up quickly get into like
Yep.
220, 200, 180 meters, that zone.
Okay,
And
great.
then just run as far as we can along the ridge crest.
Okie dokie.
Would be nice if we could have a quick look at this guy.
Yep.
Yeah, okay.
The last one, Pat. We're going to have to
Push it
start
there.
giving you an allotted amount of zooms.
Yeah.
Quota.
Yeah, zoom quota.
Okay, that's lovely.
Look at the kind of concentric circle-y stuff around it.
All right,
Okay.
that's good enough.
Come on.
Is that an anemone? It is.
No, we're cutting you off. If you lateral to your port side, you might get more tether for now.
Yep. Yeah. Like, Has the wind come up or anything? Or?
Looks like there might be some wind gusts. getting up to like 17, 18, and then dropping back down. But that's still not too bad.
Starting to move again.
Right, we just crossed 350 meters.
Woo.
Headed up. That basket star there looks like it. Yeah.
Is that a fish? Push in there, Dave. It's hiding. Camera shy.
We're not making much headway, are we? Can I ask the ship to go a little faster? More
Sure,
beans?
yeah.
Okay. Let's see if that...
Yeah, we were doing that earlier. It's fine. It's just
Yeah?
gotta keep moving.
Well, that's the mission now.
Yep.
Bridge now. Once you find a good heading, we could do 0.5 knots. Same 30 meters. 085. 085.
We have a look at this over here. Coral colony looks like.
Yes. Yep, we'll just speed up a bit.
Okay.
Thank you.
Yeah, you want to zoom in on those
Yeah,
corals?
push it
Yeah,
there.
great. Oh, they're urchins. They're not corals.
Okay. Yeah.
Very interesting. That looked like the one that got away, or different?
Ah.
Well, come
Yeah,
by
we're
there, Dave. Yep.
we're getting wind bursts, so we're
Well, anyway.
We're getting pushed away.
22
Yeah, that's
now.
okay. Let's keep moving. Hey, Kevin, look at this. Is that a fish?
Sorry, there's not much I can do right now. The ship's dragging
Sorry?
Hercules around. Oh, I was asked for a zoom on that fish.
Oh.
Might be. Oh, I got a little bit of leash now.
Yep.
Sean.
All right, Jake, want to go for a zoom there?
Yep, absolutely.
This, yeah.
So cute.
Okay.
It's a bit wobbly.
Lovely. That's 340 meters.
Chaunacops indeed.
Okay, great.
Yep.
Onward and upward.
All right, come wide.
Pulling wide.
What kind of fish was that?
I don't know, what do you call it?
Anglerfish, right? Chaunacops.
This is just a shutdown.
Seems like they're coming close together, wind and current. Should we, aren't we normally like bow 2? Is our best heading? Isn't our best heading like bow towards, bow into the predominating swell and current? Yeah. Word. Yeah, they're having trouble.
Hello, Alex.
Yep. Getting taken for a ride. We want to go the opposite way. Okay.
Okay, so we're trying to reacquire the bottom. The ship's trying to get a better heading. We've got some wind gusts that blew us off. So once the ship reacquires a heading, hopefully we'll get back down to the bottom. But if you look at high pack, you can kind of see how we just kept getting pushed. We're trying to go directly into the
East.
current and the wind, which is
We're trying to go east, and
Yeah,
the current's
trying to go
and
east.
the wind is blowing the other way.
Everything's going the other way. Well, very great.
Looks like they're adjusting their heading now.
Yeah, they're going into the current.
Okay.
As we're repositioning the ship, we have a question from the chat about, we know that we are cataloging what deep sea corals are at along this transect, and we have some beautiful video of them. Is there any other data that's being collected on this dive?
There's a whole bunch of data. We have all sorts of sensors on the vehicle. CTD, so that's conductivity, temperature, and depth. We have an oxygen sensor. We have a sound velocity sensor. Various sonars that are collecting data about the seafloor and surrounding features. And yeah, a whole host of sensors and data that are being collected throughout the whole dive.
What is this information usually used for?
Usually it aids in the habitat classification. So you want to know the properties of the seawater that you're sampling in. So what, if there's lots of corals in an area, what are the properties of the seawater? How much oxygen's in the seawater, that sort of thing. I think that's usually the main driving focus.
Thank you.
I think they adjusted the calibration of the sensor. Yeah. So who knows what it's reading? Yeah, but it's green, so.
Oh, well, that's a problem too. We've got 2 knots of current all of a sudden.
Oh,
That's new. I recorded it at 12:50, so 5 minutes ago, and it was 0.3 for the last, like, hour. That explains a lot.
is the EK80 up? Or, you know, that's usually not up during dives, right?
That's a good question. Hmm. It's different. Who to believe?
Do you want to switch over the positions source to USBL?
Yeah.
Over left bottom. I think that's just that one, yeah. Think.
Do samples ever crawl out of the bio bins? Bio boxes? Okay.
Sorry, not if they're closed. If we open the box, then yes, possibly.
Have they?
Oh yeah, definitely. Depends what it is though. Things like squat lobsters and
Sea cucumbers.
sea cucumbers, surprisingly, actually are escape
Yes.
artists.
They eject their ballast and
Yeah.
take off.
Yeah, so usually if we get something that is prone to escaping, we do not open the box again after that. I'm always surprised how fast those sea urchins move though.
Yeah. I feel like the sea cucumbers surprise me more.
Yeah.
Because I feel like the sea stars at least have like little feet.
Mm-hmm.
And I was like, okay, you have a bajillion feet, you're going to use them. Whereas the sea cucumber is basically, what, a gut rocket?
Gut rocket. Yeah.
This way? Yeah. Sorry, I think I forgot how this works. I think you can delete it directly.
Wait, which one?
You might have to switch back to the
Oh,
direct.
direct.
Yeah. I don't know why the position isn't following.
Oh, you know what? It's this one.
Oh.
There.
There we go.
Boom. Okay, I think they're
Figuring things out.
figuring things out.
Would someone in the front row be okay walking us through what's happening right now?
Sure. We are waiting for, so Nautilus is able to hold position while we're doing ROV dives using a dynamic positioning system. So it's a combination of the bow thrusters and jet pump. And sometimes if wind gets really high or if current gets too strong, we get kind of blown off station and the ship just needs some time to reacquire a good heading, which usually involves kind of splitting the difference between the current direction and the wind direction. So sometimes the ship has to, you know, move the bow to point in a more suitable direction. So the ROV team and I are monitoring the DP system up here and just seeing how they're changing it, and we're waiting for a call from the bridge to say that they have reacquired position and we can continue forward with our dive. So that's what I'm monitoring. I'm sure the ROV team is monitoring something different now that they're off the seafloor.
Yeah, we're in blue water. We're managing the distance between the vehicles and making sure they're far apart and not in any danger of getting tangled up or running into each other. And making sure we're not anywhere close to the bottom. We're going to run into the bottom. Because we have no control over the ship in a position like this. So, you know, we're attached to the ship. We have to move where it goes. So when it gets dragged off, we get dragged off. So we have to work with whatever the ship is doing and keep the vehicles safe. Huh? No, farther away.
Can I reset this?
Yep. Yeah, that was fun.
So normally you can see Hercules from Atalanta's camera, but because of, like Jake said, the inability to control where we're headed right now as the ship reorients, the vehicles are facing away from each other right now. So you won't see them in their cameras until we start along the seafloor again, or descend and then go along the seafloor again.
Okay.
How much did we need to ascend to help? with the
Actually, we didn't really need to ascend at all because we got blown off the slope.
Okay.
But if it had been the other way, we'd have to have ascended quickly, very quickly.
Yeah.
So usually we set up to do dives like that where we are on the leeward side of, you know, so the wind would blow us, wind and currents would blow us off of the slope while we're diving.
If we were going the other direction, we would not be sitting here calmly waiting for the ship to reacquire. Well, probably still calm, but quietly anxious.
What kind of work goes into designing dive plans like the one we had today?
It's a great question for the science team.
No input from us. No, that's not true.
Well, Pat chose the site, right?
Can you repeat the question for Pat?
Yeah, so we have some viewers who are wondering how dive plans are designed. For example, the one that we're using today in terms of duration, site location, what goes into planning these dives?
Sorry, you're asking me as a scientist?
I am asking anyone who contributed to this dive plan.
Yeah. I mean, you know, first of all, you know, you usually have something you want to look for or look at. So that's sort of, the basis of your planning. So for example, for this dive, we wanted to survey this, 180 meter deep ridge, but we're also interested in what's occurring along the edges of the ridge where it's going down in the deeper water. And with an ROV, it's easier to start deep and work your way up shallow than to start shallow and work your way down into the depths. So that we picked, an offshore area around 500 meters initially. We would drop the ROV in there and then work our way up the slope to the top of the ridge and then work along the ridge. Sometimes your planning doesn't work out too well. We had some strong currents and stuff, so that we started at 700 meters and we made it to 300 meters, but we're not making much progress up the slope from there. So, anyway, you start with some objective. You want to learn about something or other, and then you look at the best way to do it, whatever your equipment will allow, and then execute your plan and be flexible because things may not work out exactly like you want. We're in that situation right now where we're sitting here drifting, not drifting, but just the ROV's hanging in mid-water, and we're not seeing any bottom at the moment. But the crew is trying to get the gear back on track and down to the bottom, and if the currents cooperate, then maybe we'll be able to complete the dive. So that's about all I can say about it. But you got to be flexible, adaptable.
Yeah. Okay. Okay. One 100 meters.
100 bearing.
Okay.
30 meters.
30 meters. Speed?
0.3.
Okay.
Unless you want to go slower. Or faster.
No, we can go faster.
Okay.
I'll be up front.
Bridge nav.
And we can just stay.
We can go 0.5 knots, please.
Actually, see if I can get back out front and then come back a little bit and down, and you can turn around and face. So we can both approach looking forward and seeing the slope with the sonars.
Okay, science, we are putting in a ship move. We should be heading forward soon and getting Hurricane Atalanta lined up again as we're heading upslope.
Okay. Okay, you can yeah. Probably won't be able to just yet, but all right. Can try to turn around. Can you send the reset when you
Yep. Sure can. What's that one? Okay.
Nice.
That looks more right. Or that looks better.
Yeah.
More accurate.
No better.
More better. I think zooming through marine snow is the closest we can get to flying through the stars.
Commence light speed.
I guess it's less poetic when you think about what it's made of, but
Picking it up in the sonar.
hmm?
Coming down.
Oh yeah, perfect.
It's ahead of us.
Reacquiring.
I'm going to do another forward move. Get back to where we were.
Great. Bridge nav. Let's do another 30 meters, 100.
Samantha, you just implied about what marine snow is made out of. Could you give a little bit more context for that?
Marine snow is a delightful melange of the finest detritus the ocean has to offer. Actually, the finest detritus the surface has to offer, because it's all the bits of, you know, decaying algae and bits of food. It's plankton. It's waste products from other animals. It's anything that blows into the ocean from the surface. It's the slime and mucus that some animals make. It's chopped up pieces of jellies as they float down. So it's all the things that come from the surface and float down, and actually are quite a big source of food, both for midwater animals and also for seafloor animals. There have been some pretty incredible studies over the years, recently actually, from scientists looking at how much the volume of nutrients that benthic animals in the deep sea actually receive from marine snow falling from the surface. So,
Does the composition of marine snow change with depth?
that's a good question. I think anecdotally we find that sometimes, but it's, I think it's more about location, but that's a good question for scientists. One of the midwater animals that is a powerhouse of filtering water and filtering marine snow are the larvaceans. They create snot houses or snot palaces
What?
made of mucus that allow them to trap marine snow falling, and then they're able to filter that out and feed off of it. And in Bari, the Monterey Bay Aquarium Research Institute, some scientists there did a very cool study about larvaceans. And they found that not only were they like filtering an insane number of gallons of water a day, but their snot houses also feed a lot of creatures in the deep sea as well
Oh.
when their sun houses fall to the seafloor.
You said these were larvations?
Larvations. L-A-R-V-A-E. Wait,
So is that a snot palace that we're seeing right now?
could be. L-A-R-V-A-C-E-A-N.
Could be.
Could be. Snot house, mucus palace, you know. It's what makes a house a hum.
Sounds like a great buffet.
Bridge nav. We can add another 30 meters to 100.
Bottom in sight. Target acquired.
Target acquired.
Oh, look, it's the seafloor.
Found it.
Long time no see.
Okay. Time to get back up in the saddle.
Norbit's looking very abstract.
Is that Victor Gorgia coral there?
Looked like it, yep. Victor Gorgia.
Oh, it's raining.
Yeah, little squall coming through.
Yeah.
25 knots of wind.
Where'd you see that high wind?
Huh?
Where did you see the high wind?
I was looking at the wind speed on the
Oh, did you say 30?
25.
Oh, yeah, okay. Yeah, it seems to be gusting through. And now we're back up to a knot and a half of current.
Also, you can see it on the wire cam. It's like
Oh, yeah.
wind blowing out the deck.
Blowing. Yeah, apparently it's dumping outside. Lynette was going to come up, but she's waiting until it stops. Bridge nav. Let's add another 30 meters.
What was that? Lots of these stars.
How do you feel about me turning your aft light off? Yeah?
Shut it down.
Man, it's Sea Star City there.
Yeah.
Yeah,
The
well,
same ones we
Beautiful.
saw earlier.
Yep, 320 meters.
Wow.
It's a spawning aggregation. Just joking. But that's really impressive. Wow.
So much for Shark City.
Yeah. Yeah, haven't seen a single shark, have you?
No.
That's something. So I think we're getting into a little, little more gentle slope. Or maybe that's one reason those showed up.
We have some students from Mexico right now who are wondering where we are.
Well, we're at 320 meters off the end of the Shark City Ridge off the western barrier reef of the Republic of Palau.
Bridge nav.
What makes ridges so special to explore?
Add another 30 meters.
Well, that's an interesting question. This ridge is particularly interesting because it projects out to sea from an area that really has no other ridges. So it's unusual in that regard, the geomorphology. So that, you know, we've studied many other areas along the sort of general slope of the reef, but this is something different. So we would expect that there might well be some different habitats or some different organisms here. So it's looking for the new, you know, it's the old business of exploration. You know, if you go to the same place over and over and over, well, pretty soon you'll know everything that's there. But so you go to a new place and see how it compares, and that's what we're doing right now.
Has anything in particular surprised you about this dive tonight?
Ooh, yeah, quite a few things. I mean, it's not very exciting in terms of like finding some rare fish or something, but you know, the amount of sort of coarse and it's biologically derived material, gravel and seashells and other things that have been coming down the slopes from the top of the ridge over probably hundreds to thousands of years, and that's accumulating on the slopes. That's something you wouldn't normally see along the general barrier reef. There the sediment's much finer, thicker, and so it's just, you know, in terms of the deep slope, it's a very different environment, and we're seeing a number of different creatures that we wouldn't normally see.
Is the sediment made up of both organic and inorganic matter?
Most of it's inorganic. It's, you know, some type of, you know, usually either, you know, volcanic material, basalt, or calcium carbonate type material, which is formed biologically by corals, calcareous algae, many other types of organisms, what are called calcifying organisms. And
What are some examples of calcifying organisms?
calcifying,
Yeah.
well, stony corals, that skeleton is made of calcium carbonate, a mineral form called aragonite. Foraminifera, which are little, actually single-celled animals that have a calcareous test, a little shell they live in made of calcium carbonate. Those are animals. You have plants like calcareous algae, which have little tiny, we call them spicules, little bits of calcium carbonate material, which the plant actually secretes inside of them. So when they die, all those little pieces that are called sclerites become part of the sediment. The organism, you know, deteriorates, you know, falls apart, and that becomes part of the sediment.
Are those the same as diatoms?
No, diatoms have their skeleton made of silicon, basically like, think of it as glass in a sense. And so they do have a you know, a skeleton test, as it's called, but it's
All
made
right,
out of
keep her moving.
silicon
Let's do it.
dioxide.
All right.
Keep on trucking.
Yeah, we're up to 310 meters.
Bridge nav. Can we move 30 meters, bearing 100? Thank you.
We're making tracks now,
Thank
aren't
you. a fish down below you.
Down low.
Yeah.
Yeah.
It's like directly below you. See it?
It looks like another Synaptobranchid eel.
Oh yeah.
I like how it moves.
Cool. It's got like a paddle-like tail too. It's
Yeah.
interesting. If we weren't 300 meters deep, I'd think that was a sea snake. Just
Another fish there. Push in there, Jake.
Yep, sounds good.
How deep do you find sea snakes?
oh, well, they're air breathing, so they don't go particularly deep. I would guess,
Oh, another one of these.
you know,
Ones.
less than 30, 40 meters. I don't know the exact figure, but you know, so they're an air breathing reptile.
Hold on, wait.
We have in Palau, we only have one species of sea snake. There are other areas not so far away, New Guinea, that area, they have several species.
It comes back up.
Sea snakes are cool. The ones in Palau actually lay their eggs on land. They crawl out of the water and then they mate and lay their eggs in very shallow water, not shallow water, just islands right next to the reef. It's quite a biological event when they're out mating on the shore.
Is it a large event where there's many, many sea snakes?
Yeah, they're fairly limited areas, but yeah, there'll be a lot of them together. I've actually never personally witnessed this, but I know plenty of people who have observed it and have described it. It's been described in the literature, but yeah. Yeah, we're getting that nice little sea star. Let's see, we're up at almost 300 meters now. I like to call, yeah, if you know, you haven't tuned in lately, I like to call that the apple pie sea star because it's got these, look like 5 little slits on the top of the crust. Looks good enough to eat for Thanksgiving.
Yeah, it looks like
Yeah.
someone put a really nice egg wash on it.
Yeah.
Finally, someone who understands my desire to eat sea stars.
Cool, there's some
Cookie
nice
stars, apple
nice
pie
sea
stars.
urchins there.
Ravioli stars.
Ravioli stars.
We're getting into a very, rock right here looks very smooth. Can we,
You want to look at this urchin?
yeah, let's
Okay.
take a look
Yep.
at that. I think we're just, I would guess, looking at the bathymetric map, we're starting to come up
Ooh,
on the ridge. Just
Yeah.
the
You can, push in
bottom
there.
slope is getting a bit gentler. And can we zoom in on that?
You can push in there, Jacob.
Yep.
Get a beauty shot. Yeah, nice. Lovely.
All right, happy with that?
Yeah, that's great.
Cool, come back wide.
Hold on, wait.
We'll see what we see in the next few minutes, but you know, the rock here looks a bit smoother. Not as many, you know, indentations and little crevices and stuff. Well, I made a liar out of myself right there, but
Immediately see.
Anyway,
Nice little ball here.
so we're through 300 meters on our way up. Fantastic.
You ready for a move? You want a minute?
I think so. I think we're ready.
Okay. Bridge nav. Can we have another 30 meters, 100, please? Thank you.
It's like tabletop flat.